So in this slide, we are going to discuss about the role of single strand binding protein. And these single strand binding proteins, they are known as, in short, they are known as SSBP, single strand binding protein. Now you can observe in this diagram, when there is DNA replication, see, this helicase is moving forward, DNA polymerases, they are working together, and this DNA strand is completely twisted, it is super coiled. So if you just remember or imagine a spring like structure, if you that spring like structure is completely twisted, okay, and it has certain shape. So when you stretch the spring, there is change in its shape. That supercoiled shape of the spring, it is different when you stretch that spring. But again, when you don't apply any force to that spring, what happens? When you release the force, again the spring try to understand that spring try to regain its original shape. Okay, so the same thing can happen in the DNA replication. So how it can happen? As the DNA strand is completely folded, it is twisted around. So one enzyme is trying to untwist the DNA. So what will happen? After a certain point, the untwisted DNA will try to twist again. Okay, so to avoid the twisting of the untwisted DNA, we need the untwisted DNA, because we need the two separate DNA strands. Try to understand that the DNA structures, they are like this. We don't need such supercoiled two strands of DNA. We need the uncoiled two strands of DNA, because we need two templates. These are two our old DNA strands, and in the DNA replication process, new DNA structures, they are going to get copied from this old strands. Okay, so the untwisting of DNA is very much important for the replication of DNA. So to avoid this, when this is untwisted DNA and this is the twisted DNA, okay, so we don't want the twisted DNA. So to keep the structure unwinded, to keep the structure of DNA untwisted, another group of enzyme it works. Okay, so that group of enzyme is called a single strand binding protein. See, the name of the enzyme tells, it explains its function. That is the magic of biology, that you don't there is no need to memorize the things. The words, they magically they express their meaning to you. How it is how it is in case of single stranded binding protein. Let's try to understand role of single strand binding protein. Now see, these are the single strand binding proteins. Now this is a single strand of DNA, and this is another single strand of DNA. So single strand binding, these single strand binding proteins, they bind the DNA strand. So this double stranded twisted structure, when get in untwisted, here at a certain point, the double stranded structure, when this is a separate single strand and this another is a separate strand. Okay, so here somehow the single strand binding proteins, they get attached. Because of this attachment, what happens? It remains as a single strand. Another single strand of DNA remains as a single strand. They bind to the single stranded DNA structure. This is very much important. Okay, why it is important? Why the single stranded binding structures are important? Because if there will be no presence of single strand binding proteins, then what will happen? The untwisted double stranded DNA will after some time will get twisted, and there will be trouble in the DNA replication process, and that we don't want. So what is the importance of the single strand binding protein? Now see, these are the single strand binding proteins. Each separated strand acts as a template. Now this is one template and this is another template. These two double helical structures, they are separated and they are acting as separate template. The two separated strands are prevented from rejoining or recoiling. So that recoiling of the DNA is prevented by these single strand binding proteins. So these SSB, that is single strand binding proteins, they remain attached to both the separated strands and allow the synthesis of a new polynucleotide strands, that is new DNA structure get formed. That is, there is formation of the daughter DNA. So the fourth important step in the DNA replication process is the formation of replication fork. The point formed due to unwinding and separation of the two strands of the DNA appears like a Y-shaped fork. That appearance of the structure is Y-shaped and it is called as replicating fork. Due to unwinding of strands, there is strain, there is tension which get relieved by the superhelix relaxing enzyme. So let's try to understand this replication fork with the help of diagram. So if you can observe this diagram, see that there is semi-conservative replication of the DNA. This is lagging template, this is leading template, and this structure is the Y-shaped structure. Can you observe that? This is the Y-shaped structure. So this is a Y-shaped fork and this is called as replication fork, that you have to remember. And as there is unwinding of the DNA, as the replication fork, it moves forward. So there are a number of enzymes which work together to release the strand or to release the tension. Try to understand this point. When we are talking about the twisting of the DNA, so there is certain tension you are talking about. Okay. So for when there is unwinding, another part, this single-stranded binding protein is here and topoisomerases, they are working, helicase is working, and the replication fork, this is Y-shaped fork. But at this point, at this certain point where the replication is not occurring, at this point the DNA is supercoiled, or at this distance the strain can get increased, or the tension, the twisted structure can get again more twisted, or more the tension can get increased at this point. For the release of the that strain or that tension, supercoiling of the DNA, there is another that get relieved or that get released or minimized by a few groups of enzymes. These enzymes, they are known as super helix relaxing enzymes. So don't forget the name of the enzyme. This is very important if we are writing something about the replication fork. The fifth step which is involved in the replication of DNA is the synthesis of new strands. From the old parental DNA, there is formation of new daughter strands of the DNA. So each separated strand acts as a template for the synthesis of new complementary strand. The synthesis starts with the help of small segment of DNA molecule, which is known as RNA primer. That we are going to discuss again in the next part. We are going to talk about that. But the RNA primer is the small segment which is present. Now you can observe the diagram. The pink colored strand is the old strand on which it acts as the template on which the new blue colored strand get formed. So when there is formation of the newly formed structure of the DNA, it gets started with the segment of the RNA molecule. This is a small segment of the RNA molecule. That part is called as RNA primer. It is just like when you want to construct a wall. So before constructing a wall, we require a basement, right? So that basement, on that basement, which is the lowermost part of the wall, on that the construction of the main wall gets started. So that basement is nothing but, that part is nothing but the RNA primer. On that RNA primer, the nucleotides, activated nucleotides, get attached with the help of the DNA polymerase enzyme. As the DNA polymerase enzyme gets attached with this RNA primer. Okay, so that is the role of RNA primer when you talk about the synthesis of new strand. So the RNA primer gets associated with the 3' end of the template strand. It gets associated, RNA primer, it get associated with the 3' end of the template, that means the old strand of the DNA. Then it get attracted, the complementary nucleotides, they get attracted to this RNA primer from the surrounding nucleoplasm. These nucleotides, they bind to the complementary nucleotides on the template strand by forming hydrogen bonds, and there is formation, addition of nucleotides which are activator. We have seen that activated nucleotides which are present, which are suspended in the nucleoplasm in the first step of the DNA replication. So the DNA polymerase gets attached to the RNA primer. The nucleotides, activated nucleotides, they get attracted, and there is synthesis of new strand on the template DNA. So the next part is the role of DNA polymerase. Without the DNA polymerase, DNA replication, it cannot work, or DNA polymerase is the main role, main character of this DNA replication movie, you can say. So DNA polymerase is an enzyme that synthesizes the DNA molecules from the nucleotides, triphosphates, that is activated nucleotides. And these enzymes are essential for the DNA replication. The DNA polymerases, they are very much important for the DNA replication process. These enzymes are essential, they are very important. They usually work in groups to create two identical DNA duplexes from a single original DNA duplex. So during this process, that is formation of a newly formed DNA from the old strand, there is formation of a new strand, so that during this process the DNA polymerase, they reads the existing strand. You can observe in the diagram that the DNA polymerase is reading the information which is present on the existing strand, that is from the old strand. The DNA polymerase, they reads they read the information which is present in the form of nucleotides. And to create the new strand, complementary strand, it is very important that the DNA polymerase must read what is written, what information is there on the old strand or on the existing strand. So these enzymes, these DNA polymerases, they catalyze the chemical reaction, they increases the rate of reaction. So DNA polymerases, they add nucleotides to the 3' end. You can observe in the diagram that how DNA polymerase is moving now. So these are the activated nucleotides which are getting added. So the DNA polymerase, they adds nucleotides to the 3' end of the DNA strand, old DNA strand, one nucleotide at a time. Every time a cell divides, DNA polymerases, they are required to duplicate the cells of the DNA so that a copy of the original DNA molecule can be passed to the each daughter cell, newly formed cell. So in this way, genetic information is passed from one generation to the next generation, from the old cell to the newly formed cell or to the daughter cell. So before replication can take place, an enzyme called helicase, it unwinds. That is again shown in this diagram. See how beautifully the helicase, it unwinds the supercoil, the DNA, and the DNA molecule from its tightly woven DNA structure, or that form, tightly woven, tightly packed, twisted structure of the DNA. So in this process, the breaking of the hydrogen bonds between the nucleotide bases, it occurs. This opens up or unzips the double-stranded DNA structure to give two single strands of the DNA that can be used as templates for the replication of for the formation for the attachment of the DNA polymerase. Again, it is very much important that we need two template strands of the DNA. We need two old strands of the DNA. So now see how beautifully there is unzipping of the double-stranded structure. This is helicase. There is formation of replication fork, and after that there is formation of primer. So this another enzyme primase gets attached. It forms a base, that is foundation, as I have told that this is the basement of the wall. Now the wall is getting formed. To this RNA primer, the DNA polymerase gets attached, and from the 3' end there is adenation. Now these are the activated DNA bases or activated nucleotides. They get attached, they are added as a leading strand get formed. So the newly bound nucleotides get interconnected by the phosphodiester bonds. Now there is formation of phosphodiester bond between the newly formed DNA and the old strand of the DNA, that is template of the DNA. The synthesis of a new complementary strand is catalyzed by the enzyme, that is DNA polymerase. The new complementary strand always formed in the 5' to 3' direction, or from 5' to 3' direction. That direction is very much important because there are two strands and they are anti-parallel. So one strand is formed in a continuation or in a continuous manner, that is called as leading strand. Another in the form of chunks or in the form of small fragments, and that is called as lagging strand. So that's all for today's session. Thank you.